Okay, we'll just wait one more minute for just a few more folks to join. Thanks so much for starting the recording. Just a heads up for everyone, we are recording we are recording tonight, so just something to be aware of. If you are coming up as Virginia East, just a reminder to please go in and rename yourself. So if you find yourself on the top there, you can double click the three dots and there will be a rename option in there for you. So please feel free to put your first and last name, your affiliation, and if you'd like to include your pronouns as well, that would be great. Okay, well, thanks everyone for joining. We're happy to welcome you to the annual Coastal Forage Fish Network's annual virtual forum. This year, we're going to be talking about urban shorelines, balancing competing values in coastal habitats. My name is Caitlin Perchalski. I'm the co-chair of the Coastal Forage Fish Network, along with my colleague, Kyle. Do you want to unmute yourself and wave? Thanks, Kyle.
Hi, everyone. Nice to have you. Thank you.
So we're really excited to have you here tonight. So I'm just going to click through here, share the agenda with you. So I'll do a brief introduction today about the Coastal Forage Fish Network, and then we're going to get right into sharing our speakers' work with you. We have Mark Tagle here, as well as Jason Hall and Melinda Paulus. They're going to be speaking, Mark Tagle will speaking a little bit about surf melt story, and Jason and Melinda will share a shoreline armal removal project that they've been working on. We'll then move into annual updates from the Coastal Forage Fish members. This will include an update from NIC, North Island College, from the Mount Arrowsmith Biosphere. Oh, I always get this wrong. Mount Arrowsmith Biosphere Reserve. Oh, Kyla, I'll get it one time. And then the great folks doing wonderful work down in Nanaimo in association with BIU. We have Pacific Salmon Foundation here. They're going to be sharing their dashboard that they've developed with us. And then we have pods as well who'll be sharing. And last but definitely not least, we have the folks at Peninsula Streams and Shorelines. And then we'll be concluding the sort of presentation and learning portion of the meeting. And then we're going to flip right into the Coastal Forage Fish Network's AGM. All the folks that are here today are welcome to join us and continue on with us into the AGM if they're of interest. But for the most part, what we'll be asking for folks is who are voting members of the Coastal Forage Fish Network, for those folks to absolutely carry on with us into the AGM, where we'll be doing a quick bit of AGM business from 7:45 on to 8 p.m. So first and foremost, what is the Coastal Forage Fish Network? We're a network of stewardship groups, nonprofit organizations, and First Nations, all dedicated to the study and conservation of forage fish in BC's coastal waters. We do have some folks who join us from time to time from south of the border. So thanks to those folks for joining us. And maybe we can even expand the definition of what we include as our geographic jurisdiction. So for the most part, our focus is on the shore spawning forage fish. Examples of this are surf smelt and Pacific sand lance, who are sort of the key areas of focus for us, although we do a little bit of work with other species as well. The main scope of our role is to share information, knowledge, and resources. We undertake collective research, and we help support new groups in getting forage fish monitoring programs up and running in our region. And largely, we're looking to close data gaps and knowledge gaps around forage fish and support knowledge sharing about these beautiful, small, but mighty fish. We have about 18 different members, I believe. The number is always growing, so I'm super grateful for all the folks that are here today and for all the folks that are supporting this work. This is a list of our current members, which includes Cowichan Tribes, Esquimalt First Nation, Friends of the San Juans, the Friends of Semiahmoo Bay. We have Cayuga and Checklaslat First Nation, the Loon Foundation or PODS. We have the Malahat First Nation, Mount Arrowsmith Biosphere Region Research Institute. Thank you. That is the correct way to say that, or MABRE, Nature Vancouver, PSF, And then we have two arms of Parks Canada from different regions, from the Pac Rim region and the Gulf Island National Park. We have PSS, Project Watershed, of course, the Quatsino First Nation, Redfish Restoration out on the west coast of Vancouver Island, the Sunshine Coast Friends of Forage Fish, and the Tsleil-Waututh First Nation as well. So I just want to thank all of our members and just recognize the time it takes to participate in this and to contribute to our sort of cross-jurisdictional knowledge sharing. So thanks so much. Here's a group of us last October as we got together to do a little bit of updated training. So what do we do? A little bit more about that. So we support forage fish data collection across coastal British Columbia. That usually comes in the form of visual IDs, so finding eggs on beaches, but we also do eDNA surveys as a part of that work as well. We coordinate data collection and sharing in collaboration with the lovely folks at PSF, who have the Strait of Georgia Data Center and who are always working with us on great ways to store, share, and analyze their data. So thanks to those folks, and thanks for supporting this. We also do training and knowledge sharing. So this past year specifically, we did training for all of our stewardship groups, all the folks out in the field doing data collection, but we also did our first industry course, so training environmental professionals and other folks on what to look for in these sorts of beach environments. We also do equipment and resource sharing, so we love to share knowledge, but we also have equipment and that we can share with our groups and help get groups equipped to get out on the beaches and understand them a little bit more deeply. Here's a great map of where our partners and our members are located. So if you're really keen to find out a specific region and who's out there collecting, this is the map of where those folks are. So in the northmost of Vancouver Island, up there in the northwest coast, we have Quatsino. And then, down here in our southernmost tip, that's where we have Peninsula Stream Society. And then obviously all up the East Coast and
Oops. Well, it appears we've lost Caitlin for the moment. We can Virginia, do you know how many slides she had left, or can we dive into our first speakers?
I'm right here. I don't know what just happened. I think it's still okay. Are we still okay? Are you guys are you seeing my screen again? Is it okay?
It looks like it's loading. Yeah, we're back up. Carry on,
okay,
Caitlin.
Cool. Sorry, don't know what happened there. Let me just get back online. That was really weird. Okay, so quickly, this year's achievements have been many, and we're really excited about the group. So we continued our projects using the BC Shrift funds to support stewardship groups, as well as restoration works and our sort of individual stewardship activities. PSF launched a website and dashboard, which they will be sharing with you later tonight. We hosted an all-network training event in Nanaimo, which you see here, on field techniques, herring surveys, food dye to assist with egg finding, and also shoreline health assessments. We also developed and taught our industry professional workshop in Nanaimo and Vancouver, so we did two workshops there. We worked with our data specialist to expand the work on the habitat suitability model on the West Coast, so doing eDNA collection out there. We had multiple equipment and training opportunities. We engaged 5 new members. Shout out to the new members and thank you for being here. We also engaged 5 new affiliate members, which is great. We celebrated Surf Smelt Day on October 25th and Pacific Sandlands Day on December 11th, both of which are official days in the Comox Valley Regional District. And we also hosted several member meetings to coordinate our efforts and share with all of you folks. So these are just a quick infographic to share some of the key highlights of 2025 or 2024, 2025. So we have 28 organizations, 10 of those are new organizations. We collected 847 visual samples, 168 eDNA samples. We have 419 herring spawn observations on our INAP project. 600 volunteers are engaged and contributing 3,100 hours, which is so cool. These are our total sample results of all the samples collected. We surveyed 38 new beaches this year. And we had our official days, and then we also identified 28 new positive beaches. So those are beaches that previously hadn't been identified as having these fish present. And so 28 new beaches were identified having either surf smelt or Pacific sand lance or both this year, which is pretty exciting. And now I will pass it over to you, Mark, or actually, sorry, over to Kyle to pass over to Mark. And we look forward to learning from you.
Thanks, Caitlin. Wow, that's some really impressive stats and numbers. And thanks to Project Watershed, yourself and Virginia for sort of leading all of that hard work and herding all of us other groups and for your leadership. Now I have the pleasure of introducing our first of speakers. Just want to mention all of our speakers are coming from south of the border. And I just really want to extend that heartfelt welcome to them for giving up their time to cross that imaginary line that splits the Salish Sea, this kind of shared incredible place. So thank you to our speakers for making the time just in advance, and we're so happy to have you. But with that, I would love to introduce our first speaker, Mark Tergal, a senior aquaculturalist at the IBSS Corporation, which is kind of an interesting organization. I did a little bit of looking at it, it looks really, really interesting. Mark's work spans the intersection of ecological restoration, aquaculture production systems, and toxicology research, a really, really interesting intersection, with hands-on experience running a variety of fish species, including surf smelt. Mark has contributed to building essential laboratory baselines that inform and support field research. And today he'll be sharing some of his fascinating work with surf smelt and other forage fish. My pleasure to introduce Mark Tergal. Thank you.
Hello, everybody. Let me jump right into sharing my screen here. Share, and we'll go into slideshow mode. All right, can everybody see that? And can everybody hear me?
Sounds great, Mark.
Okay, so we kicked off. I was the senior aquaculturist for IBSS. That actually all changed on the 15th with the way our administration is going. So while I can still work with IBSS, I'm kind of out on my own because federal funding for that all dried up. But maybe they're going to use the money to help the transition to make you guys our 51st state. Who knows? But so I kind of branched off to Tagal Oceanic. We're doing all of the same stuff that we were doing with IBSS. And like I said, I still partner, and we're still kind of running through a couple contracts through them. One of the things that I had done at IBSS is we had developed 3 kind of outreach and educational programs. These are geared more towards the general public. They're designed to promote stewardship and ocean awareness. They are the Miroir of Seafood that kind of focuses on the environmental factors that, make seafood taste the way it does. Rockfish Rangers and Forage Fish Friends, which is the one that is of interest to you guys. Forage Fish Friends has been in the press a little bit lately, and I think that's how I came to your attention, was either the biographic article or perhaps from the CDOC Society. If you want to see another version of kind of this talk that I'm going to give you, I'll put the link in the comments after. But the Mass Aquarium here did a Science on the Sound. And you can kind of hear the same story, but that one also includes a really big slideshow of a really huge natural spawning event that I was lucky enough to witness last summer. So how do we get the surf smelt? That all started back when this happened, Exxon Valdez. I was attached to, well, actually when it happened, I wasn't even attached to NOAA yet, but this spill in Prince William Sound was home to a different forage fish, the Pacific herring, which there was a natural spawning and nursery in that area, as well as a really robust commercial fishery farm. So a Pacific herring was the first kind of fish that the Ecotox group, NOAA's Ecotox group, was looking at as one of their indicators of the health of the sound. And so because there was so much attention to herring, they had really kind of hammered out a spawning methodology for the fish. They had done LC50s. They were looking at sublethal concentrations and just how the oil was affecting the fish then and even now. We still go back and we look at the kind of remnant effects of what was going on there 30 or so years later. So you may be asking, like, well, what does this aquaculture guy have to do with any of this toxicology stuff? Well, my main background is in larval marine fish rearing. I've done a bunch of fish in my career for restoration efforts, for straight up production efforts, for a little mix of both. Um, but the way that I get attached to NOAA's then EC division, the latest incarnation, their ecotox division, is they like to look at the ways that different pollutants, crude oil, affects the heartbeat of small forage fish. But those guys are all chemists, or, so my role with them is providing fish husbandry, working out laboratory spawning methods, and giving them the best fish possible to then look at and, look at an example of this is what a laboratory good fish would look like versus what is showing up in polluted waters. So the first forage fish that we were working with was Pacific herring. That was because that was an economically important fish up in Prince William Sound. About 10 years ago, NOAA, Washington Department of Fish and Wildlife, the CDOC Society, two universities said, hey, let's expand herring and let's start knocking off some of the other forage fish. We've gone about as far as we can go with them. Let's start looking at surf smelt and sand lance. And so my role for that was figuring out where we were going to get the larvae from. We originally started out by trying to gather eggs on the beach, and that's all fine and good, but if you want to start with what you know is a really clean sample, they wanted to be able to spawn fish on their own so they would know exactly how old they are, kind of work out development from point of fertilization on. So that brings us to surf smelt. That was the first one we were going to try outside of herring. When they first did it, we were going to like, okay, well, we've done herring. We're pretty comfortable with that. Surf smelt, how hard can it be? No problem. We'll tackle surf smelt. So we tried to do surf smelt the same way we were doing herring. We tried capturing them the same way that we had previously captured herring. We tried teasing them open and removing the reproductive organs to fertilize the same way we were doing herring. As you can see it in the slide all the way over on the right, it just wasn't working. We spent about 2 years kind of banging our head against this, and we were figuring, well, if we just get them at the right time, or these fish just aren't ready, or we just catch them later on in the year, and we just weren't getting it. And one of the problem is when we were using kind of our beach seining method that we were doing for herring, we were getting the surf smelt just a little bit too early. The eggs hadn't fully hydrated inside of them, and they just weren't at spawning time. So kind of stepped back and we looked at this again. Is okay, so what do we know about the way herring spawn? They're spawning in the intertidal, they're slightly offshore, their eggs are really sticky, they're sticking to a bunch of stuff. And we're finding them spawning in the same waters, but not exactly the same places. Surf smelt eggs are found up on the beach. The only time we're ever finding herring eggs up on the beach is when they broke away from whatever the herring had spawned on, and those eggs got pushed up on the shore. So we started trying to rethink, like, okay, there are these slight differences in the fish, even though they're both small silver cigar-shaped bait fish. They're not exactly the same. Where they were spawning was really different. They're, kind of a rocky, sandy habitat as opposed to eelgrass was one of the big difference. And so we looked at, because when we were originally trying to even locate surf smelt, we were going kind of to these eelgrass habitats instead of fishing kind of in these rocky zones. And so we changed up the way they were fishing. We were able to catch them, but they just weren't ripe yet. Another thing that we were noticing early on is the eggs are slightly different. Herring eggs were sticky all over the place and would stick to anything. Surf smelt eggs, turns out, are only sticky at one small little spot. And so when it comes to incubating the eggs, that would become important. But it was we had gotten to the point where a lot of the toxicology folks were just saying, this is just too hard. We're just not figuring this out. Maybe we should just kind of stick with herring and not kind of move on to a new species. So we're like, okay, let's really think about this. How is this gonna come to us? Sometimes the inspiration for science comes from places you'd least expect it. So I was out surfing one day. Okay, that's not me surfing, but that is me surfing. So I was out surfing one day out at a spot over here on our side of the Straits of Juan de Fuca called Twin Rivers. It's a nice left-handed wave. This beach is also one of the spots where we would occasionally beach seine to try to capture surf smelt. But when we would do it, we were deploying with a boat and we were going far offshore and we were kind of scooping up the whole school. On this day that I was surfing, this beach is also has a really robust recreational fishery there. And they just happened to be a lot of recreational fishermen were out fishing that day. So I paddled in and I was, they were just catching tons of fish in their dip netting method. So the first guy I go up to, I say, "Hey, I'm going to ask you a weird question. Can I just squeeze one of your fish to see if it's got eggs in it?" And he was like, no. And so I was like, oh, okay, fair enough. I went back up to the car, changed out of my wetsuit, put my official fish guy hat on. I kind of walked down to the beach and I noticed these two older women down on the corner and they were fishing. And so my dad is from the Philippines. And so he grew up speaking Tagalog. I can speak food Tagalog, but that's about it. And I could hear these women speaking and they were speaking Tagalog. So I was, okay, I know, I know enough to sort of know. I know that they're speaking Tagalog. I'm going to try something. This is my in. And this is why it really helps to know how to cook if you're going to deal with fishermen. So when I approached them, I just said, "Hey, how are you guys going to cook the fish?" And they're like, kind of like, "Oh, we're going to fry it and we're going to make soup." And so, right, okay, that's it. And so I said, "Oh, are you going to make sinigang?" Sinigang is a Filipino fish soup. And they were just, "Oh, sinigang." They were so happy that I knew about their soup. Yes, yes, yes. Said, "Hey, I got a weird question for you. Do you mind if I just want to feel if one of your fish is fat. I want to see if it has eggs in it." And they're like, "Oh, yeah, yeah, no problem. Go ahead." And then they were asking me like, "Well, why do you want to do this? What is there? Are the eggs edible? Are we missing out on some delicacy?" I said, "No, no, no, no. I'm a marine biologist. We're doing a study. We want to make sure that this fishery thrives for you guys, and we want to see if there are right fish that are coming close to the beach." And they're like, "Oh yeah, no problem. Go ahead." So we squeezed one of the fish, and sure enough, she was running. She was running with eggs, and we grabbed another fish, and there was a male, and he was running. And I was like, "Oh, this is great." And I thought that's where it was going to end. I thought I was going to go back to the lab, we kind of report to everybody's like, "Hey, I found a spot. Let's try fishing here." But one of the other people fishing asked, like, "Well, how are you going to fertilize these eggs?" And this is all a true story. I said, "Well, it's since the eggs are running," I said, "Well, we're going to strip spawn them." I happen to be drinking a Pepsi that you get on the ferries over here, and so our very first strip spawn was into a Pepsi cup. And so I was just going to demonstrate to them. It's like, yeah, you squeeze the female and the eggs come out, and you squeeze the male and the milk comes out, and you kind of mix it up with a little bit of water. And this is how the eggs. That's what it'll be. And I dumped it out on the beach and left and threw the cup in the back of my truck. The next day, when I went to clean out the bed of my truck, I picked up that cup and I noticed that there were still some eggs that got stuck in the corner. I was like, well, I wonder if these guys did any good. Mind you, all of the attempts to dissect open the fish and fertilize in the past, nothing worked. We never got good fertilization. We got a couple of false cell caps, but nothing. These ones that were spawned into a Pepsi cup 24 hours later, sure enough, they were fertilized. And not just fertilized, there were a lot of them that were fertilized. If you look on the picture over there to the side with the little red explosions on them, those were the unfertile eggs. But our fertilization rate was higher than even what we would get when lab dissecting out herring. And so like, oh, we might be onto something. And not only did they fertilize, but they grew. and they grew really well in captivity. And because of the unique nature of surf smelt eggs with that one single sticky point, we were able to stick eggs onto a glass slide and monitor a specific egg all the way through its development. So this series here from hour 18 all the way to day 12 at hatch, that's all the exact same egg. We singled that guy out. We watched the development all the way through. In the summertime, we found that the surf smelt were hatching out in about 12 days. In the wintertime, it's taking them up to 20 days. So temperature is really a big factor. We also found out that no matter how cold it gets, it never fully arrests it. They'll slow way down in development, but you would always see some development every day. So we were pretty confident that it was like, okay, we think we got the fertilization thing down. And so we went ahead and published on that. And this is a poster that was presented at the American Fisheries Society on a methodology to field strip spawn surf smelt. And we were getting 93, 95% fertilization rates and then probably 50% hatch rates. So that methodology worked. At the same time we were doing this, we were looking at different incubation methodologies. Or here's just another array of some of our little guys growing there. So we're looking at two different incubation methodologies. One of them was totally in the lab and we just did it under controlled conditions. The other one is we took the eggs back out into the field and we designed these bait cups where we could bury the eggs in the sand and look at development there. And this methodology worked. So now we were able to locate the fish. We were able to find when they were spawning, and we had a methodology for spawning them, fertilizing the eggs, and incubating them. And it turns out getting the fish when the recreational people are targeting the fish is when the fish are pushing up close to shore, and that's how they were able to get them with their dip nets. And the reason they're pushing so close to shore is those are the fish that are coming into spawn. When we were just beach seining them, it was the school that was gathering close to shore, but they weren't quite ready yet. So when we waited for the recreational guys to start catching them, almost every fish that we would sample would be ready to go. It was always skewed. There were way more males than females, but we figured we'd hit all three with the reliable spawning methodology. So from that, a host of stuff came out. They were able to do land-based impacts on spawning habitat. We were able to document surf smelt development under a couple different temperature regimes, under a couple different laboratory incubation regimes. And then the whole reason was to look at studies for acidification in fish and for the ecotox guys for their pollution studies. So one of the first ones we looked at was urban stormwater and crude oil as developing heart of marine forage fish. We did surf smelt and herring side by side. And they were able to see that even sublethal concentrations of early season stormwater discharge or crude oil that had been on the beach for 20 years was still going to impact the cardiac development of surf smelt. So as another fish to be in the tool of kind of pollution monitoring, we were pretty confident that we could now add surf smelt to that role. The group that I did a lot of this work with was NOAA's Ecotox group and Department of Fish and Wildlife. Sadly, the same reason why my job changed on the 15th, a lot of the jobs for these guys changed on the 15th. The last thing that we heard was the Ecotox division has been kind of dissolved. They are currently looking for a new home, probably to partner with one of the tribes on our side of the border and just wait this administration out and see what comes of it. There was with our Merwart outreach program, that's part of the Puget Sound Restoration Fund that does that. And their oyster farmer, Josh Boma, I was out there at one of their harvest events. And this quote really spoke to me, and it talks about how things are kind of going bad right now, and they're looking bad for the US government and our ability to keep our waters clean. But if we just stay vigilant and do what we can within ourselves, even without federal funding right now, then we should continue doing that. So that's what I'm doing. I don't know how much longer Forage Fish Friends are going to be able to last. I still have a couple of grants that I was working with. We have some interest from universities that still have state funding that aren't federally affected. So we'll see how far we can go with that. So what's next for Forage Fish? We're gonna, we're like I said, we're gonna continue working on it. I don't know that I'm going to be able to do a lot of, grand, big toxicology studies, but we'll fit it in where we can. We'll continue doing the monitoring that we're doing. I noticed in one of your slides, you guys had the little blue gold panning bowl. We do the same thing on this side. So I'm working with the Clam County and the Elwha tribe to do that same beach monitoring to see what beaches eggs are showing up on. I also am doing a project with Western Washington University at their Shannon Point Marine Center, where we're taking the information that we learned on fertilizing, incubating, and hatching out surf smelt. We want to kind of complete the circle on larval rearing and get them through metamorphosis. And they want to start monitoring feeding rates using a variety of live feeds. And so then they can in turn look at feeding in different temperature regimes as we see, wintertime versus summertime fish, heat events, and our raising climate. We also want to do more of the kind of the outreach recreational stuff. Every time that we've ever gone out and collected, eggs for one of the universities we're looking at, we inevitably would have just this big public gathering around us. So kind of what we want to do is show the people how they could spawn the fish right there on the beach when they're cleaning them and just leave the eggs right there on the beach. It's not so much that we think that the eggs that they leave behind are going to save the surf smelt population there. But what it does do is it gives all of these fishermen a sense of stewardship, and it gives them a sense of responsibility to the beach and really, just to really identify and care about the environment that they're drawing resources from. And so it's simple to do. We've had success doing it. We know what the eggs would look like. So we just want to show the fishermen, like, look, you could do this. Or if you notice these signs, to get more citizen science involved in kind of doing some of this beach monitoring. A lot of our outreach and education things are geared towards younger audiences. So we partner up with some of the micro aquariums around here, like the Sea Discovery Center. They recently put in they had a salmon exhibit for a long time. And I talked them into, like, hey, everybody has seen salmon before. Let's do a forage fish exhibit. And so now there used to be kind of a circled donut where they would have salmon smolts. We have some herring that we raised, and next year we hope to have some of the surf smelt that we raise up at Shannon Point to now be part of this exhibit. We also want to expand on kind of in our Rockfish Rangers program. We have a kind of a kids book that we partner with NOAA on teaching to classrooms. I'm actually developing a similar book that's just kind of about forage fish, and it just presents kind of the importance of forage fish to a younger audience. And that has really been something that we didn't expect, but we see in all three of our outreach programs is when you target the young minds, you get them. And then they kind of come into the fold. And so I guess get them early before they have a chance to get jaded on things. The last thing we want to do is now that we've figured out herring, we're confident with surf smelt, we want to tackle these guys. These are the hard ones. We're saving the hardest ones for last, sand lance, because this is a fish that is largely unseen and spends half of its life living underground. So it's kind of, but we've talked about it. We were on the track for funding for this prior to the beginning of 2025. maybe that'll change around. Maybe we can find other interested parties in helping us out with some of this work. But what we want to look at is a reliable capture methodology for Pacific sand lance, because there is not one. We want to figure out a reliable spawning methodology for Pacific sand lance, and lastly, we want to figure out an incubation methodology for them. Once we get all of those tools in place, then we can start looking at all of the various assays we can run to learn about baseline data with what it goes to keep sand lance ticking. So that's kind of my talk. If you want to get a hold of me, there's my contact is there. I'll drop the YouTube link in the chat if you want to see a whole bunch of pictures of a really just magnificent natural spawn day. And that's that. So I'm open to any questions if anybody has any.
Amazing. Thank you so much, Mark. That was super interesting. Yeah. At this point, I would love to welcome some questions. We have a good amount of time for questions, at least 5 minutes. So if you have a question for Mark, I would love if you could please raise your hand and unmute yourself when I call on you. Or you can also pop your questions into the chat and I will ask them to Mark on your behalf. So first and foremost, Kyle, I see that you have a hand, so please go ahead.
Yes, thank you. Great presentation, and thank you for sharing that really, really innovative and cool work. You mentioned a little bit about sort of the overlap with the kind of the stormwater runoff events. Was there any kind of look at the timing and kind of the greatest times of risk for these eggs from stormwater?
Yeah, so the worst storms are early season storms. So like after you have a summer of relatively dry weather in our part of the world, you get all of this buildup on the roads. And then those first rains that we get in, October, those first heavy rains in October, November, those are the worst ones. And so when we looked at fish that were getting hit with that, especially the first 3 storms, the Puget Sound area anyway, those ones were almost as bad as a full-blown oil spill. We weren't even looking at sublethal effects. It was just LC50s across the board. It's like, all right, it's killing everything. Longer into the season, they fare a little better as most of the stuff has run off the roadways at that point. And then it's kind of proximity to where an outfall is. So we have surf smelt spawning kind of all over the Puget Sound area. There's evidence of sand lance spawn all over the Puget Sound area. Some of the things we looked at was our clean areas out in the straits and our dirty areas are in the sound. One of the things they looked at is we fertilized fish out in the straits and we exposed them to water from in the sound. Exactly what we expected. You'd see the same heart defect, even at sublethal effects. Spawn South Sound fish, put them up in the little incubators up in the straits. Those fish seem to do okay. So, location has a lot to do with it and timing of the storm. As far as where the eggs got hit in the developmental spot, that's something to look at in the future. We had always done it we would fertilize them and then do the exposure right away. But one of the things that they had wanted to look at as, if this program was still going on is an older fish more susceptible, less susceptible to those kinds of things. And the ecotox guys could probably tell you better than me, but they're looking at RNA sequencing as a method to see like when certain things are supposed to turn on, and then if they don't, when they're exposed to stormwater events or oil spill events. So.
And are surf smelt attracted to these freshwater inputs like that an outfall would have for their spawning areas?
So I don't know that they're attracted to them, but there's lots of beaches that we sample, and a lot of beaches that the eggs show up at are near freshwater outfalls. If that's something that the fish are swimming up and down the coast kind of looking for and queuing at, we're not really sure. That's one of the things that the genetics guys that I'm collecting egg samples for, we're looking at. Is it one big superpopulation in kind of our neck of the Salish Sea, or do we have subpopulations of our summer spawners and our winter spawners, and are they moving around? So that work is being looked at, but it's still kind of underway.
Amazing. Thanks, Mark. I have a question sort of about that piece of work, just in relation to all the work that's been done down in Washington and up here now on 6-PPDQ and its relationship and its effects on salmon. I'm wondering if one of the things that maybe you folks were looking at doing when you were starting to look at new opportunities is like, was there any sort of conversation around like isolating what contaminants within the stormwater were actually causing the impacts?
Yeah, so 6-PPDQ, that's the one. That's the one we were
Yeah.
going to go headlong into. All of the experimental designs were like, yeah, let's look at that. Our partners down at Washington State University in Jen McIntyre's lab, that's the main thing that they're looking at. On a little side story, when we first started doing this, I was helping them out with the pre-spawn coho mortality over here. And the way we found out about 6-PPDQ is I sat there with a DeWalt fine tooth sander, and I was actually sanding tires down to make the dust. Because we knew it was something that was coming off the roads. And we had this cocktail of all of this stuff that was coming in. And so we're like, I remember we looked at nicotine, we looked at copper, and then it was like, it's got to be something coming off the tires. And so that's what we did. We had one plot, and I came and helped a couple times, but it was all Texas that was doing this. We had one plot that simulated a road, and they just all day long they just had a car just driving in circles to wear out the tires a little bit, and then they flooded water over the surface of it, and then they're the same thing. Then they sanded the tires down, and it turns out that for coho anyway, each of those would have the same effect. And then they were able to send stuff off to the chemist, and eventually they isolated 6-PPDQ, and it's like, okay, that's the big one that's causing coho pre-spawn mortality, and that's where we're all set to start looking at it with the forage fish. That and what's it called? Dilbit. That name escapes me right now. It's the stuff that they put in pipes when they're not pumping the oil. And it keeps the pipes from freezing up. I think it's called the Dilbit solution. But that was going to be the assays that we were going to run this year if we were doing it with forage fish. So yeah.
Awesome. Well, I mean, I'm keeping all my fingers and toes crossed for a more supportive administration and fiscal situation moving forward. So I hope you guys figure it out. And yeah, like I just big hugs from up here on the north of the border. We're sending you guys lots of support as we can. So
well, what we want is in Washington, we don't so much want to take over Canada. We want you guys to take over us. Like, just expand your border all the way down. You guys could get Southern California, get some,
Washington State.
Yeah.
Amazing. Is there any other questions from the team here? We have time for 2 more questions. If folks would like to pose a question for Mark. Yeah, Kyle, go ahead. Do you have a question?
Yeah, if no one else does. I'm just wondering about the sublethal effects and what those impacts might be on survivorship, and if there's did you have any time or studies to tease those apart?
Yeah, so they haven't gone too far into it with smelt yet, but they've done a lot of it with herring. And so what they're finding is the sublethal effects, it basically gives them all heart disease. And so the herring will grow kind of all the way up, but if they're spending their early life in an area that's contaminated and they have that heart disease, their chance of reproducing are dramatically less, because those are the fish that are going to get eaten by a sea lion because they can't outrun it, or they're not going to be robust enough to make it all the way to adulthood and to spawning adults. And so they, as kind of a correlation to that, we saw that we saw that it gave them all heart problems. And you would really need the ecotox guys to speak more to this. I remember them showing me, and I just kind of like, oh, this makes sense. You look at the heart of a good fish, and it kind of goes like that. And the ones that have the sublethal effect, their heart's still, the ones that didn't get edema, their heart still formed, but its pulse was now off. And so it just didn't have an efficient heart. So you have these, whole subpopulations of fish that all have heart disease. And so they probably don't make it to spawning. And so kind of as a population builder, those fish are almost useless. And that was the one takeaway. They saw that a lot. They definitely see it in zebrafish. They saw it in herring in both up north in Alaska, and then down when we had the spill in San Francisco Bay, the Costco Busan spill. They saw it with the I think it was the herring that were down there, and there was another longfin smelt or hooligan smelt or one of the smelt down there. They saw it with them.
Amazing. We have 2 more questions too. So
Sure.
Jacques Roy, I see you have a question. Please go ahead.
Yes, very quickly. Can you tell us, Mark, how good are the populations of surf smelt and Pacific sandlands doing in your area?
So sandlats, that's the $64 question. They're not quite sure. They find them occasionally in these massive schools. They find spawns of them where they're spawning on the beach, but they're not on there's historic beaches where they know they've spawned in the past, but they don't spawn there every year. So they're not quite sure how robust the sandlands population is. One of the things that the genetics folks are doing is trying to do some population determination, because we know we're seeing big numbers of them around, but is there a subpopulation of South Sound fish versus North Sound versus Straits fish? Are those 3 separate populations, or is it one giant population? So that's what they're doing the genetics on. What I'm told is if it turns out that they're all one big population, then they're doing okay. If it turns out that there are 3 separate populations, then the South Sound one's probably in trouble. But the populations in the Straits and kind of our side of the San Juans to North Camino, Whidbey Island, those populations are doing pretty good. There's a robust recreational fishery, but not too much of a commercial fishery to speak of. There's like a couple tribal guys that fish them, but there's not a lot of pressure on them. So they're optimistic about those ones, but they want the genetics before they make a final call on them.
So this is for the surf smelt or for the sandlands?
That was for the surf smelt. The sandlands is a big question mark there. And that's one of the reasons why we wanted to start doing work with sandlands, is to kind of answer some of these questions. One, with just locating beaches that they're spawning on. We find the eggs, but we never witness spawning events. And so that's one of the things they want to start looking at.
Amazing.
Thank
you. And Ian, please, over to you. Last question.
I thought I might save you some time. Dilbit is actually diluted bitumen.
There you go.
And what they get out of the tar sands, which the people in Alberta want to call the oil sands because it sounds better, is essentially bitumen. And they add various things to it to make it fluid enough to go through pipes. So I would recommend you don't bother with that, because it's going to poison anything that comes in contact with it.
Yeah, and I think that they knew that one was going to be the bad one, and so they wanted to have like the kind of like, this is the awful stuff, and then
50,000 Canada Goose will assure you that it's not good stuff.
Yeah.
Awesome. Well, thanks so much, and big thank you to Mark. Thank you so much for joining us tonight and for sharing your expertise. It was great to hear from you, and we're just really thankful for you to come. Great. So we did originally sort of have a break here. I think it's more useful for us to answer questions. So I would just encourage everyone, if they do want a little break, to please just take it as they need. But we're going to go on to our next presenters now, if folks are okay with that. But I'm going to take a quick second to remind everyone, if they came into the meeting as Virginia East, to please just go to the very, very top of the participant list. So you can open the participant list, by the tab at the very bottom that says participants. You can find yourself at the very top. And if you could please click the 3 buttons if your name is Virginia East and not what your actual name is, and just change it to your first name and last name and an affiliation if you'd like. So thanks everyone. Over to you, Kyle, to introduce our next speakers.
Wonderful. I just want to add one last thought on that last talk, because I don't think I'll ever look at a Pepsi cup the same way. And if you could spin it, you might be able to find some sponsorship funding from Pepsi. You know, they might be into that. But anyway, next up, we're joined by some folks from the Kramer Fish Sciences lab. We have Jason Hall and Melinda Paulus. Jason is the program manager for Kramer Fish Sciences. From working with the National Oceanic and Atmospheric Administration's Northwest Fisheries Center in the Snohomish Estuary to the Estuary and Salmon Restoration Program Learning Project, his focus spans multiple deltas across Puget Sound. His work emphasizes the value of long-term monitoring and adaptive learning in estuarine restoration. We also have Melinda, and Melinda is a senior biologist at Kramer Fish Sciences, where she worked for the past 4 years. Her fieldwork spans diverse habitats and restoration projects throughout Washington state. Notably, she was of the field term during the pilot phase of the SRP Shoreline Armoring Study, Phase 1. Today, she'll be offering a field-based perspective on the challenges and discoveries made during this important study. So we're super excited to have these folks sharing their research and learnings with us. Thank you so much. Welcome.
All right. Thank you all. And quick hello to Mark again, fellow Northwest Fisheries Science Center alumni there. Melinda, you want to pull up the slide deck for us?
Yeah.
Thank you. Yeah, so thank you for having us here, folks. We've recently wrapped up phase 1 of a project, and we were asked to share some of the results with you all today. So just first, want to thank all of our co-authors and collaborators on this project, a fair number from the Northwest Fisheries Science Center as well. So just want to acknowledge them. I think we're...
Sorry.
Yep, no worries. I can ad lib. So this project was a pretty big collaboration. We had a lot of help from project partners. It was an ESRP learning project, which is a funding group in the Puget Sound tied to salmon restoration, recovery, restoration efforts generally. So this project was funded by ESRP, and we had a lot of support from our NOAA collaborators, Anna Cagley, Josh Chamberlain, Paul Cheragino from the Northwest Fisheries Science Center. They helped us with some of the sampling, and were able to leverage some of that work that we were doing previously when I was with the Science Center. In fact, same boat, if the picture comes up, I think is in the background of Mark's presentation, the stickleback. So that's kind of cool to see. So if you could advance to the next slide. Is it still stuck?
Yeah, sorry.
No
I don't know
worries.
what's going on.
Okay.
Oh,
Well, I can kind of
okay.
keep, there we go.
Okay.
Okay. There's the stickleback real quick. Okay. So just in this slide deck, we just wanted to kind of, it's a condensed version of our webinar that we gave recently. Just going to go over some basic background and our objectives, the study design and approach, and then I'm going to hand it off to Melinda, who's going to do most of the heavy lifting here. So I'll keep my talking to a minimum, and she's going to go over the pilot study results and our the outcomes of the study and how it's going to inform phase 2. Okay, next slide, please. So I don't know if this is a similar situation for the northern portion of Salish Sea, but the United States portion of Salish Sea that we affectionately refer to as Puget Sound, about 1/3 of the shoreline is armored. And as I'm sure we're all familiar with, armoring impacts shoreline processes as well as fish, particular forage fish and others. And armor removal has been identified as a priority for recovery in our region. This map shows, I think, a condensed version of 125 armor removal sites throughout the sound, and the red shows all the shoreline that's currently armored. So it's a big effort, but some substantial effort has been made. many of these projects have been completed and many more are planned. Next slide, please. As part of another project, one thing that we're seeing that seems to be a pretty consistent theme in a meta-analysis we're doing for another project, we see while a lot of projects, restoration projects in our region are being actively monitored, or as we like to refer to them for our funding agencies as evaluated, because they don't tend to like to fund monitoring directly, but we've evaluated a lot of these projects. One thing we're seeing a consistent pattern is that the nearshore projects aren't being evaluated or monitored as often as projects in our larger river deltas. Not entirely sure why this, the drivers behind this, but that's just kind of a pattern we're seeing. Most of our projects inside estuaries and deltas are being monitored, while about 50% of those associated with shoreline restoration projects in the near shore, like armor removal, aren't really being monitored. Also, monitoring typically focuses on very physical metrics and less on the fish. And when it does, the fish is focused on assemblage and abundance, kind of simple stuff. And forage fish frequently, but not as common as the salmonids and stuff. We need to start evaluating and compiling more information on fish responses for these projects. And it takes a lot of effort because a lot of the restoration and monitoring is focused on individual sites. And we don't really have a lot of comprehensive studies to evaluate fish responses at broader geographic regions. And this information can help us inform restoration planning. Okay, next slide, please. So we adopted some tools that were previously developed with the Shoreline Monitoring Toolbox. We sought to take this more traditional survey methodology that focuses a lot on discrete transects and quadrat-based stuff and is really great. But we wanted to explore the use of maybe some new technologies like drone-based data acquisition that can provide a more continuous coverage of the site and just evaluate how these methods might help us add to the shoreline monitoring toolbox and ask questions at bigger scales and kind of evaluate the strengths and weaknesses of these methods. And obviously, we're not going to be sampling fish with drones, but focus more on the physical side of things. But we're not trying to replace the tools is all I'm trying to say. We're trying to add to our toolbox here. Okay. So our primary goals said were to test some drone-based protocols and metrics and compare those to our traditional methods. As part of this phase, we developed some recommendations for phase 2, which is where we will be taking a suite of sites that we've identified and selecting a subset of those and implementing the protocol recommendations in phase 2. And see, next slide, please. I'll just kind of briefly go over. Our overall study design is what we call an extensive post-treatment design. So we require a control and an impact site. So an impact site is a place where the shoreline armor has been removed. Control would be where existing armor remains. That paired treatment and control design allows us to do differences between the sites and compare results or differences across a whole broad geographic region for sites that have been restored at different times over a broader landscape and really sort of leverage information from a broader geographic region. We also opportunistically will include reference sites, but that's kind of value-added information. We're really focused on this sort of paired sites with armor removal and existing armor within close proximity. So that's a quick overview of our study design and background on the project. I'm gonna hand it off to Melinda, and she's gonna go into some of the pilot study results and key findings.
Yeah, we'll get into it. Thanks, Jason.
Yep.
So we did 3 sites in the Puget Sound area: Boulevard Park, Cornet Bay, and Seahurst. This kind of inset here is showing Boulevard Park doing with 2 reaches. Unfortunately, we couldn't find a good reference site, but our Cornet Bay and Seahurst, we did have reference transects. And we, kind of like Jason was saying, we wanted to look at these different data types, the different specific data metrics, and then looking at the different data collection methods. Fish was looking at the beach versus like 2 field methods, beach seining versus snorkeling, and then the rest of them were traditional versus drone. So for snorkeling, we had two paired snorkelers swimming along the transect. And then afterwards, we did beach sanding because we did kick up sand, didn't want to ruin their visibility. We did this at the treatment and control reaches. And as you might imagine, each method has strengths and weaknesses. Snorkelers are, it's a little bit more low tech, it's low cost. You're able to swim around obstacles. However, you can be limited by the water visibility. Uh, Beach sailing, on the other hand, is a little bit more expensive. It requires a boat, but you can potentially get a bigger haul of fish. And also, you need to make sure that you have a good seal. You can't necessarily do it with like armoring and riprap in the way. So our results, we saw, when we say few fish, we only saw two species, and I think those were red crab for our snorkelers, and we got no size information. Even at our best site, which had, I think, 2 meter visibility, they just weren't seeing anything. Beach sailing, on the other hand, we captured a lot more fish, and this kind of graph on the right is showing a comparison between treatment and control abundances across the three sites. Asterisk, this is not statistically like sound. This is just an example that we saw like an increase in our salmonoids and forage fish. Snorkel surveys, can't do them very well. Even the things that we would be interested in, such as behavior or position in the water column, the visibility kind of didn't work out. Beach seining, we got a really good understanding of the fish composition and their fish and their size. However, the behavior in the water column, a little hard to do since the fish is in the net. But despite this, beach seining was a clear choice. You just need to make sure to do the seine when the beach is exposed to get a good seal with the net in the bottom of the beach. So that concludes our two field methods. Now we're going to look into the drone versus field sampling for these other datas that we were interested in. So we did the traditional transect field methods for each of our sites, and then we flew a drone later in the season. We did this at the treatment and control reaches. I think I already said that, sorry. And each method, as you might imagine, has benefits and disadvantages, which I'll get into with each of our different data types. So for rack, we were really interested in the rack width, composition, coverage, and then with field effort, can we do subsampling? It takes a lot of time to ID the rack composition. So we were hoping that we can try and eliminate some samples but still get an accurate representation. So this plot here is showing the rack width at the new and old rack first bit, then the control and treatment reaches at our three different sites. Cornet Bay, the tide came in a little quicker than we were expecting, so we were only able to do it at the treatment to compare the field and image, the drone, which is the imagery light blue, and the field, which is the darker blue. Old rack, we were the field was a little bit greater width compared to the drone derived width. New rack was just a hot mess. It was less than or it was greater than. And kind of with new rack, it can be a little bit more spread out. So that could be why we were having a hard time in the imagery determining the new rack. But we did determine that we can subsample rack in the field surveys, which would be great going into phase 2. Hopefully save a little bit of budget because federal is all hot mess. So from our rack, we can basically do it all in the field. But it should be noted with the traditional method, you're doing a specific point in time. You have to extrapolate to get a more better understanding of what's going on. Drone imagery, you can do it all minus composition. At least at the height that we flew the drone. I know if you fly it lower, you can maybe get a better resolution. Yeah, and I think lost my train of thought. I think that's everything. Yeah, and then rack composition we can't do from that. Yeah, can't do that. And yeah. Wood, we wanted to look at the log line width, counts for large wood and small wood. Large wood's greater than 2 meters and 1 to 2 meters for small. And this is two plots that were for one of our sites. All of the plots look the same across the three, showing the different methods of the field on the left and the drone UAV on the bottom. Logline width and large wood count were not statistically different. So either or can work for each method, but the drone imagery definitely had a harder time detecting the smaller wood counts, that 1 to 2 meter range. So that was detected to be a significant difference going forward, something to consider. So from traditional, we can do it all basically. From drone imagery, we can do it all minus the small log counts. Just a little bit too hard in the imagery. Digging into substrate, we wanted to look at the surface composition and then digging below 5 centimeters below. From drone imagery, can we determine the surface composition, which you might already have a good guess about what we found. And then we wanted to see if we could subsample surface quadrats. So on the top is our surface image. Down below is Jason actually digging, doing some hard work. This plot on the right is an example at Boulevard Park. We have our percent composition at the control, which is the top, and the bottom, which is our treatment. And then the two pairings are the different elevations that we sampled along the beach. And then 5 centimeters is below surface is surface. And I don't know about you, but this we could not determine any relationship between digging up below. So that kind of scratched out potentially only just doing surface compositions. We couldn't find any strong relationships between the control and treatment, the different elevations, or among even across different substrate classes. And unfortunately or fortunately, we need to do excavations to characterize subsurface substrate. We also took pictures in the field just to see if we can, at a later point in time, just have folks characterize the imagery. As you might imagine, smaller substrate is really hard to distinguish. You can't exactly feel the difference between sand, which is really kind of gritty, and silt, which is more smooth. So we still have that, no AI yet on that part. The feel method is definitely the most accurate. And then also like with shell hash, it can be hard to distinguish it from a small rock. So from our quadrat surveys, we can do it all. We're really good in the field. Drone composition, again, at the height that we flew the drone, it was all just a blurry mess. But from field photos, we can do the composition, but so long as we take a pre and post, just kind of knowing there might be some error associated with the smaller substrate sizes. Our vegetation, we had these questions: high tide to vegetation difference, distance, percent upland of vegetation, percent backshore vegetation, percent overhanging vegetation, the aquatic vegetation coverage, and the aquatic and riparian plants. Which ones are around? And basically, the top 4 we can do either or. Aquatic vegetation, we can do both so long as we fly the drone at a low enough tide. Eelgrass in particular hangs out at lower beach elevations. So if you fly it at a high tide, you're not necessarily going to capture that eelgrass, which would be bad. Aquatic and riparian vegetation, folks are just so much better at IDing plants, still cannot quite determine plants from drone imagery. So for the last data that we were interested in was the beach profile. And it should be noted that we have to set up the RTK, which is real-time kinematics kind of GPS systems for both methods. We're comparing the transects versus drone, and then within the drone, should we do LiDAR versus structure for motion, or SFM? Structure for motion is taking kind of ortho mosaic imagery and kind of applying some fancy modeling to it to get a rough elevation model, which can be really comparable to LiDAR. It's just that it's a lot less expensive. So here is example, the kind of showing the two different outputs. We have LiDAR, we have structure for motion on the right. The bottom is like what it actually was. But you can see here the vegetation noise and the water noise, like structure for motion really struggles. It does not do a good job penetrating through those areas. LiDAR does a little bit slightly better job. With that said, though, field transects, like we can do them really well, but they are discrete and you have to interpolate to get that full coverage of the beach profile. Drone imagery, on the other hand, kind of does it all for you, which is great. No extrapolation needed. Drone LiDAR versus structure for motion. We say yes, even acknowledging that there is some error. Since you already have to set up the RTK for the drone to know where it is, you can, for areas that might be a little bit more iffy, you can take some additional points to add to the model to help fix those like areas that would struggle with. And also to mention, structure for motion is a lot more cheaper and more budget friendly. So with that said, with that mess of data that we have, what are our recommendations and site selection for phase 2? We recommend doing both, traditional and the 21st century data. Drones are wonderful tools. They provide that continuous mapping that we're really interested in. Field surveys are still needed for that composition and IDing, like the substrate and the vegetation. But we can, with field surveys though, we can subsample, which can improve our efficiency. Fish beach staining was just the better choice. We got the assemblage, abundance, and link frequency, but since they're in the net, you're not getting the behavior or position in the water column. Again, there are trade-offs and balances, but we feel that these would be the best approach going forward into phase 2. So this is a screen grab from our report, which you can find. I'll send the link, drop the link in the chat. And basically with this, knowing how we want to approach the data collection and from the 124 sites that Jason was talking about earlier, we want to be able to identify areas that we can adequately encompass the monitoring needs that we get the I'm so sorry.
I can jump in there, Melinda, if you want.
Thank you.
It's just we're trying to, so we can't sample all 24 sites, but we have a technical advisory group that we're looking for feedback on, and hopefully we can get a group together and we can pick a subset of those that'll represent the sort of range of site ages, types of removal strategies, and different geographic regions or marine basins in the Puget Sound that we can sort of talk about things from like a broad perspective, as opposed to things that are more applicable. So say we could take the results and maybe more appropriately inform questions in the northern portion of the Salish Sea, for example. And then one thing I was going to add to here when we were closing up is just that you'll notice that the forage fish piece is missing from this. That is part of the shoreline monitoring toolbox and something we're exploring adding to the study design as well, because that piece is kind of potentially informative on different ecosystem functions and different ecological benefits and impacts of shoreline armor removal. So.
Thank you, Jason.
Yeah, no worries. You've had a long day. She's been out in the field for like 40 hours the last couple of days working on another project. So
yeah,
it's been a rough week, I know, already, and it's only Wednesday.
But yeah, with that said, you probably maybe have some questions. If you want to find out more, you can go to the Salish Sea wiki page where our webinar and report are posted. Yes, we're in the, we're in the United States, and all of our sites are located south of you guys. But if you have any like insights or are just curious to learn more, like please reach out to us, and I guess stay tuned for phase 2. Thanks.
Thank you.
Amazing. Thank you so much, Melinda and Jason, for joining us. And thank you so much for taking your time, especially when you've had such a busy week. So thanks.
Yeah.
Big shout out to you folks. We really appreciate it. We have time for 2 questions. So does anybody have a question for either Melinda or Jason? Go ahead, Kyle.
Yes.
Thank you.
Thanks so much. That was an amazing presentation. And the work you're doing is really timely for us up in BC, because we've been looking at kind of expanding as the Coastal Forage Fish Network, from looking for just the eggs and looking what other parameters we can bring in to assess shoreline health. And we've been looking at these shoreline monitoring methods and parameters that are developed down in the States and trying to adapt them up to here. So this is really timely for us, and we super appreciate you guys sharing this work that's will really help us moving forward in developing our own programs. I'm curious, one of the strengths of the shoreline monitoring piece is that community science kind of simplicity that exists. And I'm wondering if that went into your assessment at all, or the drones, if there's any kind of work to kind of make that technology more accessible for maybe not your everyday community science, but your more advancedly trained community science.
Yeah, that's a fair question. Yes, the drone data collection and to the level that you have the elevation, the structure for motion, all that kind of processing is probably not accessible to most people, because it does have to be tied into sort of that RTK network really to get a good data set from it. But it is actually relatively easy to collect. And one of the nice things about it is it provides a continuous measurement, and the data are always there. It's archived there. We envision with this next phase working with collaborators to where we can share information from the drone imagery if they collect some of the field data, or vice versa. And maybe that's a way of addressing that community aspect of it. As Melinda mentioned, all of these methods still require some component of field visit, and so there's still an opportunity to engage that citizen science. And it doesn't take that much for a team like ours, or that's experienced with the drone data collection, to go out and do that afterwards and supplement and add to that. And in fact, some of the stuff we didn't show indicated that we can actually sample less intensely with the field monitoring efforts when paired with the drone imagery. We can subsample more, which makes it easier to sample sites for citizen science with less effort. So that's a couple things I would just note on that. Hopefully that addresses your question. But yes, the citizen science part is very important.
Awesome. Thank you. Does Ian, please go ahead. One last question. And just a heads up, you're on mute, Ian. Okay.
It's not a question. For quite some time, I played flute with one of Canada's preeminent marine biologists and her husband, Tim Parsons, and Carol Lawley. And we worked out a protocol for doing snorkeling sampling. I live on the waterfront in Saanich Inlet. And we had 5 people who went out every day and did that. And we discovered it was almost impossible. We couldn't agree on exactly how to measure what people were looking for. We couldn't agree on precisely how to do the counts and so on. I was confident in what I was doing, of course, but I wasn't very confident in what the other 4 people were doing. So it sounded great for citizen scientists,
Yeah.
but it was a lot of work.
Yeah, that's a good observation. And yeah, I think we kind of echo that sentiment. Some people in our region have had pretty good luck with snorkel surveying and even scuba surveying. It's just our waters, as well as yours, can get blown out fairly easily and often. And so it just doesn't seem like a practical method to really base long-term monitoring on in this circumstance, at least. So thank you.
Great. And big thank you to you, Melinda and Jason. Thank you so much for joining us tonight, especially for this late night endeavor, especially on such a busy week. So big thank you to you guys. Thanks so much for joining us, and we're really grateful. We will be now turning over to our stewardship group presentation. So thanks so much, folks. So if those folks are ready, our first speaker will be Georgie from NIC. So I'm going to go ahead and share her presentation. Doop. And this one too. Okay. And we can go from there. Okay. I recognize you're seeing the wrong one.
Hello. Okay.
Awesome.
Yeah. Thanks, Caitlin. Good evening, everybody. I'm Georgie Harrison from North Island College, formerly faculty member, and now I'm a research associate with our Center for Applied Research, Technology, and Innovation. And tonight I'm coming to you from the traditional unceded territory of the Comox First Nation. So this is our 6th year of partnership with Comox Valley Project Watershed, working on their research on forage fish spawning habitat. And Caitlin's got to change my slides for me. I had a tech issue on my end. So next slide, Caitlin. Thanks. So our role is to provide students, volunteers, lab space and equipment, faculty support, so that's my role, and then administrative support from our Center for Applied Research, Technology and Innovation, acronym is CARTI. Next. Thanks. This year, Project Watershed assigned Team NIC 5 beaches to monitor throughout the season. So we did the qualified environmental professional survey method, which included collecting and processing beach sediment samples, and then we would take the process samples back to our lab and examine them for the presence of spawned eggs. So we had a fair number of hours devoted to this whole process, almost 400 hours. Next. And this just shows the 5 beach sites that we monitored throughout the season. So they're all located within the Comox Valley, and the distance approximately from the furthest site in the north to the southern site is about 20 kilometers. So we usually surveyed all the sites within 1 day. Sometimes it took us 2 days. Next. And a highlight was to actually see a Pacific sand lance. One day when we were surveying, somebody walking the beach found a fish and brought it over to show us. So it wasn't actually on our survey transect, but not too far away. And it turned out that the sample that we collected that day, the beach sample, had 75 Pacific sand lance eggs in it. You can probably hear my dog barking in the background. Next, please. So one thing we did this year to try and speed up the process a little bit was to actually do the sieving part of the whole process at the beach site where we were sampling. So once we sieved the sample, we had a sample that I then took home and did all the vortexing. And next. And then that sample was taken back to our lab on campus, and several different students participated in doing the egg counts. The student picture on the far left is Sam Schneider, and she did the majority of the egg count, so I definitely consider her an expert. Next. And last year, one of my students developed a technique to add just plain old red food coloring to the vortex sample, and then you wash out the red food coloring and it doesn't adhere to the sand granules. So the picture on the right, you can see that. but the dye is taken up by the eggs. And so the green circles, I can't use my pointer, the green circles are just pointing out the dyed eggs. And you can even see that without using a microscope at all. They stand out really well. And next. So this is just a picture of the eggs under the dissecting microscope. And we found that by having the eggs dyed, and some of them were pale pink and some much brighter pink, just this whole dyeing process really speeded things up because the eggs just popped out. It was much easier to identify the eggs. Next. And just a little bit of data. So of the 27 samples we obtained from the 5 beaches, we did not find surf smelt in any of our samples. We found Pacific sandlite, lance, eggs in 3 out of the 5 beaches for a total of 152 eggs. And so the beaches where we did find eggs those results are pretty typical. They're kind of like hot beaches. For the past 6 years, we've always found quite a few eggs at those beaches. 2 of the beaches, we didn't find any eggs, but you can see the number above the bars indicates the number of samples taken. So our sampling effort wasn't quite equal among those 5 beaches. In addition to the samples we collected, Team NIC collected, we also examined 21 samples that Project Watershed had collected. So we checked their samples for eggs also. And next. And this is the 5th year that students taking our 2nd year ecology course, Principles of Ecology, have had a chance to participate in this research. So one of the labs, they go out and learn how to do a survey and collect a beach sample. And then the following lab, they actually examine a sample and learn how to recognize spawned eggs and do an egg count. So this gives students a chance to participate in local relevant research. And it's also a great way for me to recruit them to volunteer for the rest of the field season, which many of them do. And last slide, I think. Just to acknowledge that we had 9 biology students work on the project this year. And in particular, just a shout out to Sam Schneider. She did most of the egg counts, and this is her 4th year working on this project. So we're lucky to have that consistency. As I mentioned, CARTI, our Center for Applied Research, Technology and Innovation, found funding to pay Sam to work on this project. And then Garnet Martins is our senior lab tech, and he makes sure we have lab space and troubleshoot some of our equipment. And then a final shout out to Virginia from Project Watersheds for her advice and ordering equipment and helping us schedule our working plan. So really appreciate the help from everybody at Project Watershed. And that's it. Thanks.
Awesome. Thanks so much, Georgie. And I'll just add, I didn't provide this context at the beginning of our presentations, but we're going to hold all the questions for our stewardship folks till the end, and we'll have time for questions after all the presentations. So at that, I'd like to pass it over to the folks at Pacific Salmon Foundation to present about their dashboard. Big thank you, Georgie.
Thank you so much, Caitlin. Hi, everybody. Let me share my screen. Can everybody see that?
Yep.
Okay. Well, thank you for having us. This is just a short introduction to the data center and our collaborations with the Coastal Forage Fish Network. And so for those of you who might not be familiar, especially coming from south of the border with the Pacific Salmon Foundation Marine Data Center, and my name is Paulina, Data Integration Specialist. We aim to be a one-stop shop for data and metadata. And so through doing this, we have a website that anybody can access. And most importantly, we have a data portal. And this data portal, which I'll show up later, is a compilation of metadata records that we have created. And by we, I mean mostly Terry and Ben, my teammates and coworkers, through almost over 10 years. And it's just collated information, websites, resources, and data about salmon and everything that affects salmon, their ecosystem, and their habitat. Now, that being said, the CFFN, so the Coastal Forage Fish Network, for those of you who don't know, is a collaborative initiative that focuses on research monitoring and conservation of forage fish species along the Pacific coast. And PSF has supported this effort basically by providing a platform through our data portal to store the data that they collect. And we're very big on open access, and so we make that data publicly available through the Marine Data Center. So this collaboration of ours aims to facilitate the efforts of monitoring and conserving forage fish populations, which are crucial to the marine food web here in British Columbia. And so recently, in the past few months, we have actually been collaborating with this network to create a centralized website, which is not quite public yet, but we are doing just the final touches. Any feedback is always highly welcome and usually integrated. So anyway, so our website includes information on forage fish, information on CFFN, some pictures of the volunteers, and a dashboard that we've created together. So if you click open full screen, it'll take you to our full dashboard. And this dashboard is really showcasing the survey effort by the volunteers and the organizations that work as part of this initiative. And so you can see the green bubbles really just showcase the number of times volunteers went out there to conduct surveys. And then if I turn that off, you can see the points show every place where there were positive egg detections, and they're color-coded by species. And so if you zoom to a specific area, you can actually see in the graph the number of times those eggs were detected and what species they're associated with. The dashboard also provides the capabilities of filtering, and it has a table associated with it that actually showcases. So if I open it, all the years from the beginning of the initiative, the number of surveys that were completed, the eggs that have been found, the total number of beaches where there have been positive detections, and the beaches that had first positive detections in that year. And so it's very comprehensive, and the reality is that the goal of it is to showcase the effort, the incredible effort that all these organizations and volunteers have worked on through the years. Now, it hasn't been perfect, so some hiccups that we encountered. Terry, my coworker, he really, along with great collaboration from CFFN, they work together to just QA/QC the data. As some of you have said, citizen science isn't always perfect. And so he addressed some positioning issues of the lat and longs of the points. He really put some thought into how to best standardize the data collection, just so that there would be less uncertainty and error in the lats and longs that were collected, because we had some points that were just a little bit random and all over the place. And so he came up with this nominal system where you basically just through the name of the beach you get the collection point, and so or the point is automatically assigned through like a nominal system. And so what we have added is just lines on the map that show the beaches that have been surveyed, just to kind of like, since the points have been standardized, you can also see what area of the beach was covered. And so if you have any questions about that, we'll be more than happy to talk about our methodology and how we address it further too. And the last thing I'll talk about today, this is a short presentation, is other ways in which we have supported this initiative is we've created story maps that describe the efforts through our map catalog. And I just want to showcase really quick those metadata records that I was talking about earlier. They're all in our resource page. So this is what our data portal looks like. And so here is where we will be including the points of the map. We just very recently finalized them, so we'll include them here for everyone to access. We include the latest data set and some of the protocols that have been kindly shared with us. So as I said, our goal really is to support the program to help make the data openly available and to really just help in showcasing the huge effort that this initiative has taken and all their findings and all the good stuff that they're doing. So that's it for us, and thank you for giving us a space to show the work that we're doing with you.
Awesome. Thank you, Paulina. We're so thankful for the work that PSF is doing. Yeah, the work would be so much more challenging without the support we get from you folks. So thank you so much. And with that, I'm going to pass it over to Sydney, I believe, from PODS. So please go ahead, Sydney.
Awesome. Thanks, Caitlin. Share my screen here. Okay, hopefully everyone can see that.
Yep.
All right, so my name's Sydney Flemmerfelt, and I work for the Loon Foundation as a biologist assistant. And today I'm going to be talking about our forage fish monitoring within the Seychelles Nation Swia and Tla'amin Territory. It's out of the way there. Okay. So the Loon Foundation was established in 2002 as the Ruby Lake Lagoon Nature Reserve Society. For anyone who doesn't know about the Loon Foundation, I'll just do a little bit of a background here. We changed our name to the Loon Foundation in 2002, and we started monitoring for forage fish spawning beaches in 2017 as part of our Pender Harbor Coastal Waters Monitoring Program. So here's where we are located within the Salish Sea. That's Pender Harbor. And in the last 2 years, we've expanded to Pell River and Texada Island. In 2017, monitoring started with just our 4 core sites, so that's what we built off of. And in 2019, we started using a blitz method. So when we first detected eggs at our core sites, we would monitor our blitz sites, which were our new beaches, and that increased the probability of finding new beaches. In 2023, we expanded into the Upper Sunshine Coast, and over time, we have identified 20 spawning beaches within our regions. So here's what a typical spawning beach might look like. And here's some photos of our processing, our vortexing there, and of course, our Pacific Sandlands eggs. So here are our Pender Harbor sites. You can see egg presence indicated by the yellow stars and egg absence by the blue dots there. So we've surveyed over 50 sites since 2017, and of those 50 sites, 13 have been identified as forage fish spawning beaches, all of which have been Pacific Sandlands eggs, except for one lone surf smolt egg that we saw at Baker Beach, which don't know where that guy came from. And here are our Powell River sites. We've surveyed a total of 19 sites in the last 2 years, and 7 of them have been identified as forage fish spawning beaches, all of which were Pacific Sandlands. So for us, generally, we note our first spawning within the last 2 weeks of November. But this past season, it was a bit later than normal. We did have quite a few pretty crazy storms in the last little bit of November and early December, which may have impacted this. But our first recorded spawn this last year was on December 6th, so it was nearly 2 weeks later than what we've previously seen. And just a little bit about our how we classify our beaches. So for our stable spawning beaches, that just means we've had consistent egg detection over multiple years. For us, this is Smelly Goat Beach, Sunset Cove, and our 2 Vaucroft sites. Declining spawning beach, of course, is just a spawning beach that has shown a decline over the years. For us, Martin Cove, you can see we had quite a few eggs identified in 2019, and then we haven't had any detection since then. And for Baker Beach, we had spawn present from 2017 to the winter of 2021, and the number of eggs observed started to decline in the winter of 2020, and no eggs were actually found after 2022. The back shore of the beach was hard armored in the spring of 2019, so this may have had an effect on that. We're not too sure. And along with those, we've got our variable spawning beaches. So variables detection some years, but not others. And then our no detection spawning beaches. So suitable habitat, but no observations of actual spawning. And then putting this all together, we've surveyed about 70 beaches, and 20 of them have been identified as positive spawning beaches. However, due to some logistical constraints, capacity, weather, many of these sites were surveyed maybe only once a year or during a single month each year. So forage fish spawning is spatially and temporally intermittent, meaning the absence of detections doesn't necessarily indicate a true absence of spawning because we may have just missed them. There also seems to be a significant variability in egg abundance between sites and years, even if the habitat seems suitable. And then, like we saw this year, storm surges, wave activity, and substrate scour can disperse or completely remove the eggs from a spawning area, making detection more difficult, of course. And all of these things together underscores the need for long-term monitoring to accurately track spawning trends, assess habitat stability, and evaluate the impacts of shoreline development and climate change, which are some of the many reasons why we're very excited to be part of the CFFN and work with all these amazing groups. And that is all.
Awesome. Thank you so much, Sydney. And thanks for that summary. I couldn't agree more. And we're so grateful to have you folks contributing to the network. So thank you so much. And like I mentioned, we'll hold questions and then we'll go straight into our presentation from PSS.
Awesome. Thank you.
Thank you.
Can everyone see that? Let's see.
Not quite in full. Yeah, it's not presenter mode yet, but we can see it.
How'd this work out? Yeah.
Click on, slide and then.
Am I missing it? There we go.
Oh, I think this, there you go.
Here we are. Okay, sorry about that. Yeah, my name is Kyle Armstrong, the Executive Director of Peninsula Streams and Shorelines. Presenting with me today is Chloe Kramer, the coordinator of our beach program. And yeah, just before we get started, just a huge shout out and thank you to all of our beach program volunteers who just continue to put up amazing numbers and are just an amazing and really qualified and competent group of volunteers, community scientists who are doing and gathering an amazing amount of data Rather than giving kind of an overview of our program this time around, we're going to be presenting on one specific site, the Nymph Point Park Shoreline Restoration Project, which is a partnership project between Project Watershed, ourselves, the District of North Saanich, supported by Project Watershed's Aquatic Ecosystem Restoration Fund funding for the CFFN, and it's a demonstration restoration site. So without further ado, Nymph Point is a small park and the aptly named adjacent beach, Pocket Beach, Nymph Point Pocket Beach, is the study area. It's backed by a park, as I mentioned, as well as to the northeast a marina. So the backshore is basically two property holders, the Oak Bay Marine Group Marina and the District of North Saanich's Nymph Point. We chose this area because our forage fish monitors have basically revealed a pattern that North Saanich, and in particular CM Harbor, is a hotspot for surf smelt spawning, and it's also a hotspot for coastal development. We don't have the linear kind of hardening data that the States has, although Pacific Salmon Foundation is working on making it public, which will be really interesting to see when that becomes available. But if we did, this whole area would be quite red in terms of shoreline alterations and dredging, hard armor shorelines, and just lots of development. So it's a key spot for us in terms of the ecological and cultural values of the site and area, and one we honed in on. Some of the site conditions, this is the northeast end of the beach, and what we refer to as kind of the degraded end of the beach. There's a couple of features I'd like to highlight. There's a rock wall that was built by the Yacht Club, the tenant of the Oak Bay Marine Group there. And so they built this rock wall to kind of stabilize their back shore. And as a result, a lot of the good kind of forage fish materials have been lost on this beach, and you get kind of as well as some of the rock armor debris has kind of filtered through the rock wall and almost polluted the sediments with the kind of hard angular blast rock. So we've kind of lost a lot of the sediments, and as well as kind of gotten this pollution of blast rock, non-native rock on the beach as well. Partially, the area has an industrial use at this end as well. The Oak Bay Marine Group has their yard. There's a kind of a boat facility, and there used to be an old boat ramp here, and the remnants of that still exist, as well as, that backshore area. There's almost entirely 100% invasive species, with very little tree and overhanging vegetation. There's also a remnant creosote log structure that exists that continues to kind of pollute the beach and sediments there. And as I mentioned again, the large angular substrate and kind of these marine glacial clays that aren't suitable for forage fish spawning. On the other end of the beach, on the southwest end, some of the features down here, we have a totally different story in terms of the physical conditions. We have an eroding backshore, slightly accelerated by kind of informal pathways that are kind of accelerating this erosion. There is some protection. There's a layer of boulders along the toe of this bank, but not a full-scale rock wall. There are overhanging mature native trees, which are really great for shading and large wood recruitment. As those fall onto the beach, they'll provide nice habitat on the beach. The backshore, thanks to the Friends of North Saanich Parks, who actively control the invasive species of the park and of the beach, is quite healthy in terms of the marine riparian composition. It's almost 100% native plant species. And also this has all kind of led to really good sediment. So the backshore is eroding and nourishing the beach naturally with some really highly suitable sediment sizes and abundance for surf smelt spawning. And with that, I'm going to hand it off to Chloe, who can speak to some of the sampling that's gone on this site and what we found in terms of the use of the site by smelt for spawning. Chloe.
Cool. Yeah, thanks, Kyle. As we've mentioned, Niv Point has this really interesting gradient of spawning sediment and spawning habitat, moving from that southwest reference site up towards the northeast kind of degraded end. We see this gradual loss of that coarse sand pea gravel mix that smelts like to spawn on. The sites that you see on the map here are our designated forage fish spawning sites that our amazing volunteer community members perform year round. We've been monitoring this site or this beach for spawning for a number of years now, but mostly focused at this southwest reference end. But in light of some restoration plans that we have for this beach coming up in the fall, which Kyle will touch on, we've been a little bit more interested in what's happening at the northeast degraded end. In terms of spawning, I'm hoping to kind of quantify that a bit better. So over the past couple of months, our volunteer teams have done some double sampling events at this beach. So basically sampling site 1 at the southwest reference site, as well as site 5 at the northeast degraded end, as a way to directly compare both egg abundance and survivorship at each end. Next slide. Yeah, and these doubling sampling events have yielded some pretty interesting results. So our first happened in January. We saw site 1, our reference site, have just about 1,200 eggs. Oh my gosh, excuse me, about 3% dead. So pretty good survivorship at that end. And then our degraded site, about 200 eggs, 65% dead, so definitely higher mortality rate. The next month, we saw actually a near double amount of eggs at both sites compared to January, which is interesting. Reference site had about, almost 2,500 eggs, only 2% dead. So again, really high survivorship. Whereas our degraded site had 400 eggs, 15% dead. And then just earlier this month in April, similar pattern, although we are starting to see spawning kind of taper off as we head into warmer weather for the spring and summer. Our reference site had about 300 eggs, 30% dead, and the degraded site only 10 eggs, 40% dead. Next slide. If you're wondering why March was missing from that, it's because we did a bit of a different event for this month. Instead of our regular volunteer team going out and sampling, we actually had a biology course from the University of Victoria come down and do a bit of a blitz sampling across the beach. So in addition to sampling both that reference site and the degraded site, they also sampled 2 other sites in between to see if that gradient in spawning existed, which we can see from the numbers, it roughly did. Basically, overall, at our reference southwest site, we're seeing higher abundance of eggs, lower mortality, whereas at that degraded site at the northeast, we are seeing lower abundance, but much greater mortality. And I will hand it back over to Kyle to talk about our restoration plans.
Excellent. So with the restoration project team, we engaged DHI to conduct a meta-ocean study and look at the oceanic conditions, and provide a scope for restoration. And one of the things that came out of that meta-ocean report was an area for, or a plan for nourishment, beach nourishment. And this plan focuses on nourishment at this northeast end. And so using, modeling the sediment from that southwest side, recreating a sediment mix that mimics that, and placing about 900 tons to basically try to recreate the conditions of that site further along. And there, so again, we're taking this kind of sediment, trying to recreate those conditions on the northeast end. We'd also like to try to curb some of the erosion of the backshore through some bioengineering techniques. So we don't want to stop erosion completely. We do want to protect some of the coastal midden material exposed. And what we plan to do is kind of cap that with our nourishment material, placing that kind of beach nourishment sediment mix on top, along with some soil, and planting it, and hoping those plants can help stabilize that back shore, as well as provide and slow the kind of failure of those trees, as well as provide new tree recruitment. So if we can plant some trees in and amongst that, and have kind of the next generation of trees coming up on the back shore to replace those that are imminently falling. We'd also like to create a formal access pathway that doesn't require kind of crossing the midden site. And so we're exploring different options right now with the restoration team and engineers. We'd also like to remove this creosote ramp. So that's part of the restoration as well. That will be removed and hopefully recycled or reused, but otherwise disposed of. Yeah, and right now we're just working through the permitting phases as well as the community outreach phases. On the left there is the design foray that we had with many different kind of groups coming to participate and discuss what a viable restoration plan would look like, as well as a photo from a recent Earth Day beach clean with the Oak Bay Marine Group staff, as well as our volunteers, where we removed a couple 100 kilograms of marine debris and a couple 100 liters of small foam bits and a small dory. So yeah, with that, thank you to Project Watershed, our project partner, as well as the other project partners, and the Pacific Salmon Foundation for funding a lot of this monitoring work over the years, and our community scientists who continue to build a really robust data set that is informing our restoration and conservation work on the coast here. Thank you.
Awesome. Thanks so much, Kyle. And thanks to everyone for sharing. I'm just going to wrap us up here. Unfortunately, we ran a little over time, so we don't have too much time for questions. So if anyone does have questions, feel free to pop them in the chat, and we will be sure to ask them to our speakers and then circulate that to the registrant list, those answers. So if you do have any questions, feel free to pop them in the chat or email us with those questions. We'll make sure that they get sent to you. So thank you so much, everyone. If you'd like to, please fill out the Forage Fish survey. You could take a little snapshot of the QR code there. And I believe Virginia has just popped the link in the chat as well. So there's the link in the chat for the form that you could fill out. So I want to give a big big thank you to all of our financial and in-kind contributors. A big thank you to the BC SHRIF granting team for supporting this work. And supporting the restoration work. A massive thank you to the volunteers who are on the ground doing this survey work and collecting this really important data set, and all of our community supporters and stewardship groups. So big thank you. Please take a moment to fill out the feedback survey. And if all the voting members for the Coastal Forage Fish Network could please stick around for the AGM. We'll try to do it as quickly as we possibly can. So thank you so much. If you're not a member of the CFFN, feel free to pop off now, and just thank you for joining us to learn more about our favorite small fish. Thank you so much. Okay, folks. Welcome to the Coastal Forage Fish Network Annual General Meeting.